Okay, so let's get started, and we want to look at circulation and respiration. And they're, of course, very intimately linked because, well, first of all, why do we breathe? Why are you breathing?
You say so we can live.
So you can live. Do you want to elaborate a little bit?
So at least, to release.
Do you remember this old equation from your last semester? And this will tell us why we breathe and also why we need oxygen. So energy, actually there's ADP coming in, I didn't balance the equation, but this is what we refer to as cellular respiration. So this is what is going on within the cells, and we utilize that oxygen to get some energy from our food, and here's the energy that's put out. Obviously this is not a balanced equation. And then we give off, as our byproduct, we give off some water and carbon dioxide. And I wouldn't really call it waste, so we'll talk about some of the functions that water and carbon dioxide actually have within the system. But why, so cellular respiration is going to be this overall equation, okay? And we breathe in and out so we can circulate the oxygen, and so we can get rid of the carbon dioxide. So that's why we need to kind of breathe in and out. So why do we need oxygen specifically? Go back to what you learned from 101. So we start off, and here we'll just do a more elaborate look. Right? And then we break it down, remember, during glycolysis. This is all your 101 stuff, right? To pyruvate, to acetyl-CoA, which is fed into the citric acid cycle or the Krebs cycle. Right? And what gives off from the Krebs cycle? What comes off? I know there's some waste and stuff, but we have some energy molecules. And what happens with these? Where are they fed into? That electron transport chain. All right, so they're fed into, you're gonna have to do it on this side, right? Ran out of room on this side. Okay, and then they're fed into that electron transport chain. And then at the bottom, the final electron acceptor. So here's the electron transport chain. And you studied this last semester, so I won't go into all the details, but the final electron acceptor of the electron transport chain is oxygen, right? So if you don't have oxygen, you don't get the electron transport chain, you don't get the Krebs cycle, you're going to only make about 2 ATP, whereas this whole process, you're going to make 36, 38, depending upon the types of cells. So your production of energy is significantly halted and not enough for you to function. So why do we need oxygen? It is the final electron acceptor in the electron transport chain, and we need to breathe, and we need to have organismal respiration to distribute the oxygen that's needed to all the cells where this is going on. And we also needed to remove the byproduct, carbon dioxide, from the system. Okay, so let's look at different types of organisms, and all of these are these are animals, and they're all going to need to breathe. Now, if you're a unicellular organism, you don't need to distribute the oxygen throughout your body. But if you're a multicellular organism, you're going to need to do that. And so here's a different evolutionary trends. Here you can see some parapodium in these marine worms, which are going to filter out the oxygen from the water. And here you can see a little bit more advanced. Here's in a crayfish, or if you look at a lobster, some type of crustacean. If you break it apart, do you ever see those gills underneath there? Anybody like to eat lobster? No, I do, right? So those are the gills under there, and the water with the oxygen is feeding in them, and the gills are pulling the oxygen out of the water. And here you can even just see a starfish. Now they don't have sort of one area like that. If you took a cross section of it, you'd see that some of these tiny extensions are really gills that are really taking oxygen out of the water. There's 2 feet here they're showing that helps the starfish kind of walk and stick on to the rocks. And here you can see it in an insect, and in the insect you have these little tracheal openings here where the oxygen is going to come into this way, and then it'll be distributed throughout. And here you can see a bird. Now birds, do you think they need a lot of oxygen and a very good system? Yeah, I can see you nodding. So it's very interesting. We breathe in and out, right? And so there's kind of a pause. Birds are up there. They have to stay light. There's not as much oxygen up at higher elevation levels. So they actually keep the oxygen, the air, going through the system in a unidirectional manner. So if you look at their lung system, they also have posterior and anterior air sacs. So here you can see anterior, meaning closer to the head, and here you can see posterior, closer to the rear. And so what happens is, and if you look at it, so the air is going to come in, and then it continually feeds around and out. Really good system for absorbing, for getting a lot of oxygen from a low oxygenated area. Okay, so here, using the trinkets. We talked about last time this counter-current exchange. And we said that opposite flows in particular areas can have a really good benefit of some physiological function. And we looked at it before. We looked at putting arteries and veins next to one another and circumventing the heat so that they didn't lose the heat to the exterior environment when you need to keep the heat. And here's another example of this counter-current exchange. So if you're looking at a fish, and here's a fish and there are the gills. And what they do is that it's set up very ingeniously. So the water is coming in across the gills one way, but the blood is flowing the other way. And this allows for the maximum transfer of oxygen into the blood. So let's just kind of look at it. Remember, these are diffusionary processes, and it's going to go by the partial pressure, the amount of oxygen that's in one area to the other. So if the amount of oxygen is high here, it's going to go to where it's lower. So let's look at it here. So the water's coming in, very, very high oxygen levels over here. But the blood also has a very high oxygen level because it's flowing this way. But it's still going from an area of higher oxygen level to lower oxygen level. So it's still coming in. And even when you get to very, very small quantities of oxygen that are in the water that have gone through this and had a lot extrapolated, it still is able to pull it in because look, the blood over here is very, very deoxygenated. And so you can still get a forward flow of oxygen from the water into the blood. Whereas if it was parallel, if they were going in the same direction, not just parallel, but actually unidirectional both ways, you would be coming in, and then at a certain point, probably around midpoint over here, you might end up getting backflow, that the oxygen going the wrong way, or you would be sort of saturated at too low a level. So does everybody see how this way you can pull and pull and pull more and more oxygen out of the water? So it's a very good system. So let's take a look at our system. And so here is the structure of our respiratory system. So we can either get air in through the nasal cavities or the mouth. What might be some of the advantages of breathing in the nose versus breathing in the mouth? Yes, you do. I don't know what that was. Okay, well, if you breathe in your nose, how do you think it's protecting you or helping you at all? What do you have in your nose? What?
It's filtering stuff.
Okay, so there are filters here, right? Little hairs, little filters, little mucus. So that's one of the things that's happening. There's also these bumps and folds that you can see in the nasal cavity. So not only to collect things, but if you're going to be taking in, it's a cold day out, right? And you're going to be taking in oxygen, that's going to be a big stressor. You're going to be taking in that cold air, that's a stressor. So it kind of warms the air as it's coming into the body. So it warms it filters it, and then it's going to be coming down and through. And if you look over here, the pharynx is another name for your throat. That's the appropriate name for your throat. So this time, last time we talked about digestion, we wanted to go through the esophagus. This time, no, we want it to go through the trachea. So the air is going to come through the trachea. So if you feel your neck over here, do this, that's your trachea. What does it feel like? Is it smooth? Does it have bumps? Yeah, it's very bumpy, right. So what are these bumps? These are rings of cartilage, okay? And it's going to keep it open. Remember, if you don't have oxygen for about 4 or 5 minutes, those cells are going to die, right? So you want to keep this open as much as possible. And so these rings of cartilage are going to keep it strong and open. So there. But then as you go down in, you're going to see that it branches off. So from the trachea, it's going to branch off into the right and left side, these bronchia, and go into every side. Now what begins to happen to those cartilage is now it's getting smaller and smaller, okay? And so they're sort of, almost plaquing, but not a full ring. And then it gets even smaller and goes down into the alveoli. So why do you think you're going to lose this very, very strong, big cartilage as it goes down into your lungs?
Do you want them to be able to expand?
Okay, so lots of reasons, right? You want them to be able to expand and contract. So you want that flexibility, and inside the lungs, there's going to be those fiber tissues that are going to allow, those elastic tissues that allow you to do that. What else? What is going to be happening in these lungs? What do you need to happen here when you get way down in the bottom where the alveoli are? What do you need to happen? I'll give you a hint, there's squamous tissue lining it. Whenever you hear the word the squamous tissue, where it's nice and flat. Exactly. So you're going to need gas exchange. So you don't want it being blocked by all this cartilage and things like that. So it gives it a lot of openness and a lot of protection. You can expand it and you can get this appropriate gas exchange. Now higher up in here, you're also going to have it lined with cells that are going to have cilia and produce mucus. But you don't want those big thick cells down in here, right? You want those squamous cells so that you can get that exchange. And so you have other types of cells that are sitting in here, immunological cells that are there to fight infection for you. Yes?
So my question is, so when you get bronchitis, like what causes that, and then why is it so difficult to get rid of it?
Okay, so bronchitis is an infection in the bronchi, okay? And so the infection went down lower. It's not, down in here. A pneumonia would be harder to get rid of, but it irritates it terribly. And if you take a look at someone who had pneumonia or a very bad respiratory infection, and you looked at a cross-section of their slide, you could take a look and you could see that there's cilia damage and surface area damage, and it's highly irritated. So that's why even after the infection is done, sometimes you'll still cough and it'll still hurt and things like that. So it sometimes takes a while for these things to heal as well. Yeah. Did you have a question? Just resting. Okay, sounds good. All right, so just to also take a look, this is where the gas exchange is going to occur. So it is high, there's a lot of capillaries that are going to be surrounding the alveoli. And it's going to be bringing that deoxygenated blood, that blood that has carbon dioxide that was given off by the systemic cells of the body to get rid of that carbon dioxide. And you can see it changing over here, they're showing it red, and that's because the oxygen that's in the alveoli are entering the circulatory system now. So at the site of the alveoli, you're getting rid of the carbon dioxide and you're bringing in the oxygen, and it's an extremely rich area over here. Okay, so here you could see it's a very dense bed of capillaries. So squamous cells lining the alveoli, squamous cells lining the capillaries. So you can get this maximum exchange of what's going on. Okay, so let's look at actually the mechanisms of breathing that help you do it. We are what we call negative breathers. Some other types of animals, like frogs and things, actually force the air down into their lungs. Well, they also have cutaneous, get air through their skin a lot. But what we are negative pressure breathers. So what we do is we increase the space of our lungs so that it causes negative pressure and the air just kind of fills in and comes in. So it does this by two different ways. Your rib cage goes up and down, and your diaphragm goes up and down. So when you want an inhalation, which is very active, inhalation is active, your rib cage goes up, right? And your diaphragm goes down. So now what you did is you increased that thoracic space and you caused a vacuum, and so air's going to be rushing in. Now, when you want to exhale, and you can see this diaphragm, so the inhalation contracts down, diaphragm goes down, ribs go up, okay, making a bigger chest. Now when you relax, you get an exhalation. So the diaphragm relaxes up, making that a smaller area. The ribs go down, they kind of go down. And so this whole thoracic area is smaller and air is going to come out. So just normal resting breathing, normal relaxation breathing, it is active for an inhalation, but it is relaxative for the exhalation. Now, that doesn't mean if you're doing more forced breathing, okay, so you're exercising and you need to take in more air, but you need to blow out more air. So that can be more controlled too, and sometimes that way it's going to be active. Okay, so let's look at what is controlling this breathing, because the breathing is going to be really important for a number of reasons. One, to get your oxygen levels up. We also use breathing to help maintain the pH in our blood. So that's another thing, and that's why a lot of the regulators that are in the blood are actually pH monitors that you have. So here's looking at it. Your normal blood should be about 7.4. Okay. Now, if you're running around and doing things and getting very, very active, what happens is, and if you remember last semester from respiration, cellular respiration, you're going to be putting out a lot of carbon dioxide, which in the blood can turn into carbonic acid, making your blood lower. You also might be putting out a lot of lactic acid, so that's going to lower the pH as well. So you want to kind of maintain it at 7.4. So there are receptors in the body that go, okay, and here they are, carotid artery and in the aorta, which is coming out, and there are sensors, and they will sense the pH. So a lot of times what your body's looking for is carbon dioxide levels, which will turn into pH levels. And then at this point, it's going to be sending messages to the brain. And it's typically in that brain stem where there's this cerebral spinal fluid, and in the brain stem, which has these autonomic mechanisms. It means it happens automatically. You don't have to think about, oh, carbon dioxide levels are low, my pH is low, I better breathe more. No, it happens automatically, and it's in the brainstem here, medulla oblongata, and there's actually a little additional regulation up here, which is the pons. So if you take anatomy and physiology, you get a little bit more detail. But brainstem, medulla oblongata, pons, that is going to be regulating the breathing. And so what's going to happen is it's going to send messages to the chest, and it's going to say, okay, diaphragm pull down, ribs pull up, and let's get more inhalation. And then what that is going to do, the blood CO2 levels are going to fall because you're breathing it out at a much more rapid pace. You'd exercise, so you're taking oxygen levels, but you're also breathing out more. And we'll get that pH back to its normal level, which is about 7.4. So here you can see it's this nice negative feedback control system that keeps us in this homeostatic range, and we want it about 7.4. Let's just look at the circulation that's going on, because it's very intimately related. Circulation and respiration are extremely interrelated. So let's take a look at this. So here you're looking at the lung. This is the alveoli in the lung, that little bottom part that you have. And when you're breathing in the air, you can see that the air has a very high oxygen level and a lower pH level. So what's going to happen because of the differentials, since the blood here has lots and lots of carbon dioxide, more than the surrounding air or primarily the air that's in that alveoli at that time, so the carbon dioxide is going to leave the blood and you're going to exhale it. And the opposite is true for oxygen. Oxygen is going to be much higher here than in this blood that came, back from the systemic cells. Remember, those systemic cells are pouring in carbon dioxide in it, and they've used up their oxygen. So if you're looking at this, the oxygen levels are low, so partial pressure of oxygen is going to be higher there than here, so it's going to go in. So we're going to go through the heart, oxygenate in the lungs, and go through the heart. And then we get to the systemic cells of the body, and the systemic cells of the body are doing this. They're doing that, and so they need oxygen. They are low on oxygen because they've been doing this. They've been doing cellular respiration. They don't have much oxygen, so they're going to have to pull this oxygen in. Low levels of oxygen here, higher levels in the blood, goes into the systemic tissue. We'll call it body tissue or systemic. And then over here, because of all that respiration that's going on in the cells, there's a buildup of carbon dioxide. So that's going to go from the higher level of carbon dioxide here. It's going to be fed back into the circulatory system, back to actually the right side of the heart is typically, it's deoxygenated blood. And the left side, remember, this is not your orientation, this is your patient's orientation, right? So this would be the right side, and this would be the left side. Okay, so the right side of the heart then goes to the lungs for, to get reoxygenated, and the cycle starts all over again. Does that make sense? Maria's got that? All right. So let's look at the magic molecule here that carries that oxygen around. And so here you can see this is a hemoglobin molecule. Anybody know where this is found? Where in the blood? Is it in red blood cells and white blood cells and plasma? Yes, red blood cells, also known as erythrocytes. So here you have a hemoglobin molecule, and you can see each hemoglobin molecule, it actually has quaternary structure, which remember from 101, primary structure is the order of the amino acids, and secondary structure is that local bending. Tertiary structure is the whole string of that peptide molecule sort of wrapping around, and quaternary structure meaning that it's interacting with more than one of these long peptide molecules. So in each hemoglobin molecule, you're going to have 4 hemes. And if you look at the center of the heme, there's iron in the center of each of these hemes. And the iron is where molecular oxygen attaches to. And the reason why we say molecular oxygen is because oxygen, you know, if you look at it, it's not happy just one oxygen molecule by itself. So oxygen molecules kind of travel around in pairs. And the hemoglobin molecule can hold that molecular oxygen to it and circulate it through the blood. Now, carbon dioxide doesn't compete for this site at all. Okay, so carbon dioxide, it has a much more complicated system. There are greater numbers of things that carbon dioxide does. There's a few side groups that it could hang on to, but carbon dioxide is also within the plasma, which is the liquid portion of the blood. And it doesn't just stay as carbon dioxide. It can turn into bicarbonate ion. It can turn back into carbonic acid. And so it's in many different forms. So oxygen is primarily here attached to the iron of the heme. Carbon dioxide, when it comes in, it can go into different forms. And we said that it has other uses too. So carbon dioxide, if you think about it, it's not just a waste product, but the blood system utilizes it as a buffer. What's a buffer? So yeah, in a general sense. And what about on a chemical level? What would a buffer be?
Keeps the pH.
Yes, right. It helps to maintain the pH. So if you look at carbon dioxide, it goes into this whole system of changing its molecular structure to carbonic acid and a bicarbonate ion. And what it does is if you get too acidic, it will actually take in the extra acid, those hydrogen ion groups, so that the blood doesn't have all that acid in it. And then if it goes the other way, it can release it. So there's a number of different transformations that can occur. So yes, it is our waste product, but it's also really important for buffering as well. So I want to point out something else. There's another molecule that sometimes will go right over here, that we don't want to go there. So that is carbon monoxide, right? CO. Carbon monoxide has an extraordinarily high affinity for this iron over here. In fact, it's 200 times higher than oxygen, and we don't want carbon dioxide there. So that's why in all of your houses and all of your homes, you have a carbon monoxide detector, and you should check it and make sure that the battery's okay, particularly when you turn on and off the heat and you know, in the season, because it can come from, you know, gas, whether it's the car in your garage or inappropriate, you know, giving off by your furnace. So there's a lot of really important things, and it is so dangerous because it will push away 200 times more than oxygen. So you get asphyxiated. And so that's why you have these carbon monoxide things. So if you're ever in a house and you feel that there's, you know, that this person may have succumbed or has fainted from lack of oxygen because there's carbon dioxide, get them outside right away. You can give them, you know, mouth-to-mouth resuscitation. You want to try and get as much oxygen in there as possible. And, you know, they might even take them to the hospital and put them in this hyperbolic chamber, which is basically forces some oxygen under pressure so that way it can replace that carbon monoxide. Kind of similar to, you know, if divers get the bends and things like that, they have to force oxygen in. So it's a really, really, really important thing. And so make sure when you go home, all your carbon monoxide detectors are on. I had a student, it was a couple of years ago, and she told me a story that she got home and her sister was passed out. The family wasn't good, and she saved everybody. So I thought that was pretty cool. All right. Let's take a look at what we would call the dissociation curve. So if you go back to the structure here of the hemoglobin molecule, you notice, as we said, that it has 4 attachment sites for oxygen. And what happens is that once you attach one to oxygen, it is easier for oxygen to attach to the other ones because it causes that molecule to kind of change its conformational shape. So it's easier. So if you take a look at what we call this oxygen hemoglobin dissociation curve, it isn't straight like this, right? You'll notice that it gets actually easier to get the oxygen attached or detached as you go up into the higher levels. And here it's actually showing you, that here's low levels of oxygen, maybe because it's where your tissues are exercising and lungs will actually be higher. So this just gives you an association. So you can see this also shows you how the oxygen would be given off. Now here, it's showing you that hemoglobin molecule, this hemoglobin molecule here, has a slight conformational change depending upon the pH of the body. Okay? So if you're exercising a lot and you're putting out a lot of carbonic acid, the shape is going to be slightly different. So, and the way that the hemoglobin molecule gives off or holds on to that oxygen changes. So look at this, at 7.4, it's holding on to it tighter than at 7.2. See this shift over here? That shift is called the Bohr shift. And it just shows you that it's a little bit different on different pHs. So why is this important? What might this do? Well, you'd say, when you are exercising and your pH is just a little bit lower, right? You want to deliver more blood to the tissue, maybe not hold on to it. And so this gives you a little bit more, oxygen to give off, because you want to bring more of that oxygen, not just holding it onto the hemoglobin, you want to deliver it to the tissues. I want to show you, so here's looking at that again, here's looking at that Bohr shift. I also want to show you one other thing. Maternal and fetal hemoglobin are a little bit different in shape, okay? Just a little bit different in their conformation. And so if you took and did a dissociation curve with fetal hemoglobin and maternal hemoglobin, you would see that the fetus has a higher affinity for oxygen. So why might this be really, really important? Why would this be important? So you're looking at the placenta. Where does the baby get the oxygen from? Yeah, the placenta, right, from the mom's placenta. So the placenta, the blood isn't running in from one to the other. There's actually, so you could think of it as like, this is mom's blood in the placenta coming in, and this is the baby's blood. So there isn't really an exchange, but there's diffusionary surfaces there. And so if the baby has a higher affinity, if the baby's hemoglobin has a higher affinity, then the oxygen is going to go from the mom more easily to the baby. So you get the development, of the baby that's more appropriately. You don't want it to be equal, you want it to be as efficient as possible, and you want the flow to be this way. So it's really important that the dissociation curve for fetal hemoglobin, it's higher, its affinity for oxygen is higher than the mom. All right, just open it up as quick and easy. Did you take the quiz?
No, I just got
Okay, go outside quickly. Hey, you are lucky. 1 minute later, and we better. All right, so you're gonna come in the back door. 1 minute later. All right. Okay, so let's go over these questions, and it also gives you an idea of like what kinds of questions will be, and you know, on the test on Monday. So for the test on Monday, I want you to understand what was digested in each of the parts of the digestive system. So as a sample, I gave you one here. What type of food is digested in the stomach? Which of those? Proteins, exactly, okay. And what type of feedback systems keep animals in dynamic, steady states?
Positive.
Negative. Something gets too high, you lower it. Something gets too low, you raise it. So what about a positive feedback system? It's what? Right, so, and it feeds, it cascades up, and it spirals up, so you want some sort of job to be done, whether it's birth, you know, getting that baby out, that oxytocin increases, the contractions increase, and everything keeps going in this increasing modem until the baby's born. And we'll see when we look at blood, actually the process of blood clotting, homeostasis, keeping your blood in you, also has to cascade up, and we'll also see it when we do immunology. Okay, so flies and birds have wings, but different evolutionary paths created that wing. So this would be an example of convergent evolution. They had totally different evolutionary paths, and then they converted on the same thing. Now what you will see, particularly when we do environment, is actually divergent evolution, which we're going to look, where you end up with different structures to solve the same, you take the same structure and it turns into different things, like the arms can turn into wings and maybe flippers. So that would be an example of divergent evolution. What you saw there was convergent evolution. Okay, flat epithelial cells that line capillaries and also the lungs, the alveoli of the lungs. Which are they? Squamous, right? That's squashed squamous cell, and that allows for transport. So when you're looking at those different, epithelial tissues, think about the function. And if the function is increased passage of gases and things, then you might want to think about squamous. If it has to produce lots of stuff, then you might want to think of columnar. If it acts as little bricks, then you might want to think of, and makes tubules, then you would think of what? Yes, cuboidal. Good. Okay. In which system do signaling hormones circulate in the blood and cause changes in the target cell? The endocrine system. Good. So we'll be doing a whole extensive review of the endocrine system. And what is the other signaling system that you have in your body? And we'll go over that later, that we compared. The nervous system. So that goes directly. Yeah, does someone have a question?
So can we flip-flop that?
Can you? Yes, possibly. A question might be.
Point.
Right. So there might be a question with which the answer could be the nervous system, but that would be going through, nerve lines and, electrical right, you can flip-flop. But for the answer to this question, that would be that. Okay. All righty. Oops, you had that.
Yeah,
You had that while we were talking.
Oh my God.
All right. It wasn't a clue that we were reviewing it, that maybe you should hand it in. No. All right. I'll look over it. Okay. So the next one, the structure of an organism and its study. So now we're looking at structure. Anatomy. Good. What if I said the function? Physiology. So I could say it the other way. Physiology would be if I asked for function, if I asked for structure, that's anatomy. And of course, they're intimately related. Okay, where are blood cells? Where are blood cells produced? In the bone marrow. Good. So that's going to be the bone, B. Okay, and here's where I sort of gave you two questions, but we'll just do the first one. Okay, what is the function of intestinal bacteria? Okay, they produce vitamins such as biotin, vitamin K, and other B vitamins. The other ones are not appropriate. They don't make it slippery. They're not there for no reason. No, they're there. They have a very important mutualistic symbiotic relationship with us. Okay, here's one you're not getting credit for, but you know what? It might be a good question for the test on Monday. So let's just at least look at the answer to that. So a number of enzymes in the body, including the digestive enzymes, are going to be made in an inactive form, so they can be produced at one location in large quantities and then delivered to another area without digesting your body along the way. So what do we call that? Zymogens. Very good, zymogens. Okay. And where does most digestion, nutrient absorption, and water absorption take place? Small intestines. Good. So when we looked at all those different things, when we were going through what's digested in what area, we know that the mouth had starch. We know that the stomach had protein. But once you get into the small intestines, some of the enzymes produced there, and mostly produced from those zymogens that are coming from the pancreas, everything is going to be broken down, digested there. And so in addition to them being digested, they're absorbed. And even though one of the major goals of the large intestine is to reabsorb water, because the surface area of the small intestines is so long that actually most of your water is reabsorbed by the small intestines. All right, let's look at the next one. In which scenario would you or your doctor be most concerned about buildup of plaque in your arteries? And I gave you got your blood tests back from the doctor, okay, and he's looking at them with you. So which one do you think would be the most, concerning? High LDLs and low LDLs and low HDLs, or high LDLs and low HDLs, low LDLs and high HDLs. So which is the bad cholesterol? LDLs. So when you have high LDLs, that's the bad cholesterol. We talked about how that is bringing it, from the liver back to the circulation. And then you have low HDLs. Well, that's the good kind. You want higher levels of that because that is taking the cholesterol back to the liver for reprocessing. So the answer there would be B, high HDLs and high LDLs and low HDLs. Okay. So let's do a little recap before we get into the next part, which is the circulatory system. And so now we can help you kind of fill out some of those charts. I also skipped over one of the things that we do in the respiratory lab, but I think I'm just going to mention it now anyway because it's in your documents at all. So we need oxygen. It is the final electron acceptor of the electron transport chain. Without it, we don't get enough energy. And so let's just kind of go through the sheet together so to make sure that you have all the answers. So give examples of different types of animals that obtain oxygen. And so we looked at some of these different animals. We talked about, gills, different types of gill structures, and also tracheids that are in insects. And we also looked at this wonderful rotating, circulating air system that birds have. So that way there can be a continual flow of oxygen and throughout the system. Okay. We also looked at, this is a very important concept, and you'll see it again and again, which means I will ask you about it again and again. And so this is the concept of countercurrent flow. And here's another example of it, and we showed that if the flow is in opposite direction, countercurrent, it allows you to do certain functions. And in this case, in the gills, it is where the water's going one way and the blood is going the other way, it allows you to pull out more and more and more and more of that oxygen. So it's a very efficient way for fish gills to get as much oxygen as possible. Okay. We described the structure of the human lungs. I mean, you know, I just want you to, you go through it, you should know the functions of each of the parts. I do want you to know how that these are reinforced. And another thing just to kind of mention, these are in separate pleural cavities, which is really sort of like a redundant or backup system. There's a lot of redundancy in life just to protect yourself. So what happens if you have an injury or a stab wound here, right? You may lose the function of this lung, but, you know, maybe cover it, hold it. But if this lung isn't affected, then you have at least, you know, the ability to get oxygen through that other area. Okay. Thank you. You can keep that.
Oh, okay.
Sure. All right. So one of the things I didn't mention, but on the bottom here, in the alveoli, there's a substance produced called surfactant. Sort of makes it kind of slippery. It reduces surface tension, and it's really important. So if you have some surface tension when those exhalation occurs, I mean, you can test with a, you know, some paper or something. There's a slight attractive force. So if I had an exhalation and the walls of the cavity slightly attracted each other, and there was nothing sort of smooth like soapy there, it might stick. And so surfactant is really important for this proper functioning of the alveoli, particularly on that exhalation. And in newborn babies, if they're premature or something like that, their cells might not be producing enough surfactant. And therefore they say, you know, the lungs aren't ready. And one of the things they're looking at is, does it produce surfactant? So they would have to give them, that we now have like big ones that we can give. But surfactant can actually be, you know, if somebody is on oxygen for a long period of time or has other, you know, respiratory issues that cause those cells that produce surfactant to not work properly, it can make breathing very difficult as well. So that's another thing that's really important. So surfactant helps reduce that surface tension and allows the lungs to work properly. Okay. What I didn't look at, and I was going to save it just for the lab, but as long as it's in here, and somebody said, well, you know, we didn't go over everything, so I'll just go over it now. So one of the things was the different lung volumes that you have. So if you're looking at the lungs and you're breathing, so let me just kind of show you what's going on. Of chalk here. So this would be kind of time, and this would be the volume. Okay, going from 0 up. Okay. So when you're breathing, it's not like your lungs are totally collapsed or anything. You're going in this nice area, sort of the rise and fall of the chest. So you're here in this area, even though there's more space in your lung at the bottom, or you can fill it up more at the top. So if you're doing resting, this is what we call the tidal volume. And you could think of the air coming in and the air going out. So that's normal resting breath is your tidal volume. But on top of that, let's say you were in a race and you wanted to go to the other end of the pool. You take a giant inhalation. You know there's more space in your lungs. So from, let's say, up here, this is tidal volume, okay, tidal volume, and you take a giant inhalation. There's more space in your lung. So that's what I put down as that first one, the inspiratory reserve volume. It's there if you need it. It's your reserve, so you can fill it up. Now you don't always fill it up to to the gills, I should say, but we don't have gills. You don't always fill it up to the top, but as your activity is going up and you're moving around more, you may need more oxygen, so you may be going higher and higher in. So that maximum space over here is your inspiratory reserve volume. Okay. Now, if you breathe out, so let's do a normal breath. Okay, and now see if you can fill your inspiratory reserve volume. We're gonna be doing this in the lab, so go. That's a big space, and it's bigger for men, actually, than women. They have a much bigger inspiratory reserve space, and that's because they have a lot more muscle that they have to feed oxygen to. But still, there's this nice big space. Okay, so now bring it back down. Okay, now at the bottom of your breath, you can blow out, right? Right, okay. So that is your expiratory reserve volume. So there's some here at the bottom, expiratory reserve volume, and I put it on the sheet over here, expiratory lung volume or reserve volume. It's there if you need it. You're running out of breath, you're under the water, oh my gosh, you know, you want to go down and you want to use up more of that air that was just sitting in there to begin with. So these three volumes together, okay, is your vital capacity. This is what you need to function. This is what's there for your reserve and is available for you. And it changes. This is at rest, tidal volume, more room, less, more room at the bottom. Now there is some space over here, which is your residual volume, okay? And that's at the bottom. That's not usable space. That is keeping the system open. So in other words, when you breathe out, you don't collapse all your alveoli. You don't break down, the things that are in here. So from the time that you took your very, very first breath, the initiation of your very, very first breath, you filled up that reserve volume. So if they found a baby and they weren't sure if the woman gave birth to it and it died, or it was a stillborn, they will look for that reserve volume. Okay? And if the reserve volume is there, that meant that baby took a breath. Okay? So that is there. Now it's not usable. You can't use it in your vital capacity, so that's why it's not there. Now when we have lab, probably not this lab, probably the next, we're going to see what everybody's vital capacity is, and then we're going to chart it and see if we actually get, some sort of a curve where the bigger people in the class are actually using less per gram than the smaller people in the class, which is what we saw when we were looking at the elephant and the mouse. The mouse had that very, very quick respiratory system, and that elephant per mass didn't use as much. So we'll take a look at that in the lab. We usually do okay on those results. Okay, so you should remember the position of the lungs, and I'll ask you that. What is it? Oh, there's that. Position of lungs. Inhalation. Where are the lungs? Up or down? Where are the ribs? Up or down? Inhalation, ribs up. Diaphragm contracted down. I know you think if it's contracted, it's going to go up, but actually it's contracted down, making it big. And exhalation, the reverse. Lungs, ribs go down. Diaphragm then will relax up. So it relaxes up. Okay. And what do we call this type of breathing? Yes, exactly, negative pressure breathing. So it's just, some animals, as we said, pull the air in. So this is negative pressure breathing. We looked at how breathing is controlled. We looked at the signals. So really you're going to get, carbon dioxide levels are really going to be looked at first because they create carbonic acid, pH sensors. So if you can't breathe, it's that buildup of carbon dioxide that causes that inhalation. And it's also true, like when a baby is being born, the initiation of that first breath is going to be from that buildup of carbon dioxide as well. Not the need for oxygen, which does come in secondarily, but it's actually that buildup of carbon dioxide that does that. Okay, we looked at the partial pressures. We looked at the fact that you always go from a high concentration of that particular type of molecule to a low concentration, and we followed it through the systemic cycle. So take a look at this, when you're reviewing it. We looked at, I'm trying to do it in order of this now. Yeah, the structure of a hemoglobin, just to help you fill this in now. And so we noticed that there were 4 hemes. The iron is where the molecular oxygen fits in. What else can compete with molecular oxygen to bind here? Carbon monoxide, right? Carbon monoxide. So 200 times more affinity from carbon monoxide than oxygen. So what about the carbon dioxide? How does that fit into this? How is that transported? Do you remember?
It's more complicated and it converts in different forms.
Yes, it is a little more complicated because it's going to be floating a lot of it in the plasma that it's going to go. There are some side places that are going to be there as well. It does not attach to the heme, and it can change, and it also acts as a buffer to keep the blood at 7.4, which is normal blood pH levels. All right, so we looked at this Bohr shift, right? We looked at the Bohr shift, and that is a change in the affinity of hemoglobin to oxygen depending upon the pH. So you get this change, and we also mentioned that there is different affinity for fetal hemoglobin and maternal hemoglobin. Fetus has a higher affinity, and so you're going to get the oxygen at the placenta going from the mom to the fetus. Okay, one thing we didn't look at is diving animals. Now, some of these animals can stay under the water for extremely long periods of time. How do they do this? They have a number of different mechanisms. So they can change metabolism. They also have, in addition to hemoglobin, in their muscles, they have myoglobin. A lot of them, we have some of that too, but they have large amounts, and that can hold additional oxygen as well. So here you can see these seals that can stay under for very, very long periods of time. There's also, humans get a diving reflex. So does anybody ever hear the saying, if somebody was drowning, they're not dead until they're warm and dead? Did you ever hear that saying? Okay, what that means is, particularly, it happens more with kids and cold water. So if somebody drowns, you'd say 4 minutes, they've been under, they're not surviving anymore. But there is this diving reflex that we kick into, that some people will kick into. And your metabolism is going to go way down. And then when you bring them up and you try and revive them, if you can warm them up, they might start breathing again, and give them CPR and everything. They might start breathing again. If you bring them up and you say, oh my gosh, they're dead, they've been down there for a long period of time, and don't try anything, that, they might have had a chance. So once they're warmed up again, if they're still dead, that's going to be it. So that's also because of our evolution and diving reflex, and so it's really interesting. Okay, so what we want to do now, and I don't have the handout for you because it's online, and I will make sure that I get it to you later. We have, actually we don't have much time, do we? All right, so instead of starting this, I will start it next time, but please read ahead. Please read about circulation, and we're going to go over the blood and the circulation and how that works, okay? Yes?
So those